Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Happy New Year. We hope you had a restful holiday season. On our end, we're excited to be back with brand new Dr. GPCR podcast episodes. Before we dive into this episode, we are excited to launch the Dr. GPCR University program with the goal of providing courses and content on all aspects of GPCR research to support our community. To kick off this new initiative, we partnered with Dr. Terry Kinakin, who will be teaching a course entitled Applying Pharmacology to Drug Discovery starting February 8th. The course will be held every Thursday through February 29th, starting at 10 a.m. Eastern Time. Spots are limited to 20 in total, and the deadline to register ends on February 1st. If you register and join us, you'll get 4 live Zoom sessions with 1-hour lectures and 30-minute Q&A each time, a 30-minute one-on-one meeting with Dr. Kinakin according to his availability, access to the cohorts group in the Dr. GPCR ecosystem, where you can find Dr. Kinakin himself and your classmates. You'll get access to the course materials and recordings. You'll get a completion certificate signed by Dr. GPCR and Dr. Kinakin. And if you'd like to take advantage of this, we're also giving you a 1-year Dr. GPCR Ecosystem Premium membership, which is a $250 saving, which gives you access to the Dr. GPCR Ecosystem contents and perks, including our forums, private groups, and to all ecosystem members. And this way you can revisit the course contents at any time and take advantage of visiting the other course contents later on. To get more information and register for the course, join the ecosystem for today for free. What are you doing April 22nd and 23rd? Join us for the inaugural Endocrine Metabolic GPCRs meeting taking place in Birmingham in the UK. This event will bring together scientists from both academia and industry to exchange data, technologies, and ideas in the field of GPCRs in the context of endocrine and metabolic receptors. We have a special offer for the Dr. GPCR community. Visit our website to get more information. The key topics covered at this meeting will be the latest GPCR signaling methodologies, metabolite sensing GPCR biology, and novel endocrine mediators and GPCR activation structures. This meeting is organized and we're partnering with Biospecifica to bring you this meeting. This meeting is also endorsed by the Society of Endocrinology. Mark your calendars again for the next season of the Dr. GPCR Symposia, and mark your calendars for our first event of the year, which is March 15th. This first symposium will cover the topic of GPCR activation and signaling. We will have talks, and we will also be accommodating posters on Zoom in breakout rooms, so stay tuned. If you'd like to give a talk at any of our events, or if you'd like to be my guest on the Dr. GPCR podcast, please email us at hello@drgpcr.com. And now let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and I'm very excited to have with me Dr. Alix Rouault. And I did it right this time. Hi, Alix. Welcome back.
Thank you.
How are you? How are you doing today?
I'm very good. The end of the day, about to go in the weekend, so can it be better?
Exciting. And thank you for joining me on the podcast. Before we hit record, we were just talking about how much fun we had on the Dr. GPCR monthly newsletter video edition. So this is a part 2 kind of, or repeat, and this time we get to focus on you. So Alix, please introduce yourself.
So my name is Alix Rouault. I am a postdoctoral fellow at the University of Michigan Life Sciences Institute. I am in the lab of Roger Cohn. He is a big melanocortin guy. He's the one that cloned it. And I'm actually working on melanocortin now, but I'm still grabbing onto my favorite accessory protein to GPCR.
Oh, great. And how did you end up in the position that you're in today?
So are we starting from the beginning or
Yes.
just okay?
No, go back, or wherever you want to start. I will take you back if need be.
So I've always been passionate about science. When I was little, I remember I have my best friend that is now in Canada, Romain Villot. I was very, very like, we were entertaining ourselves with microbiology at first. His parents bought him a small microscope that you couldn't see anything through it, but you could see some amoeboids, and other like small organisms. Couldn't go down to microbes, but we could see like a little bit of microbiology life. It was very, very nice. And from there, I always knew that I wanted to be a scientist of some sort. I had no idea what kind of scientist I wanted to be. And we had this great path to science in France. It is a technical training. I don't know the equivalent that would be in the US, but uh, what's right? Oh, it's high school actually. It's during high school. It's a technical training during high school that allows you to experience in the lab. We did a lot of biochemistry, microbiology, uh, molecular biology too, actually.
That
Um,
young, well, that young, that's interesting.
yeah, yeah, and that was. I mean, that was the basic, right? We were doing. What was my favorite part in this training was actually the microbiology, because they were giving you a um, bacteria and you didn't know what it was, and you had to seed plates and medium and have to figure it out what kind of bacteria it was. So you had to do the grams, check
Mhm.
whether it was negative or positive. It was very nice, very nice like education. It was like mini investigation on microbiology. It was super cool. So I did this through high school, and then I went to the university, and it got interesting around the equivalent of Master 2, Master 1 in the US. And so I had to do an internship in a research lab. I did my internship. So the college I went to was Bretagne Occidentale. I don't know how to translate that in English.
I don't know, but anyone who wants to figure it out, I think they can try and Google it. Although it's gonna be difficult to write it down.
The anagram is UBO.
Okay.
And so I went through I had to do an internship there, and I chose a research lab, a fundamental research lab, and I ended up not being on fundamental at all and having to work on it was a compound they were developing for a skincare product, I think.
Mhm.
And but it was awesome because on the morning we were doing abdominal surgery, and then in the afternoon we had the layer of fat and skin, and we were extracting keratinocytes from the skin graft that we took on the morning. It was very fun uh, to do, and then during that time I actually met someone else from another unit. I was a smoker at the time, reformed.
And
And
it's on record now.
yeah, oh, definitely. And I was smoking at the bottom of the building, and there was this guy that I didn't know back in the day, and he was the director of another unit, and we ended up, talking a lot. And at some point, he asked me what I wanted to do, and I'm like, I don't know, probably I want to go into research and do a PhD. And he recommended me to one of his friends. I thought he was a student at the time. He was actually never a student. They were friends together. And it was Julian Sebag. Um, he was just opening um, his lab at the University of Iowa. It was in his first year, and he said, like, do you want to like get in contact with him and see if he would be interested in having you for a PhD? I'm like, well, yeah, why not? I mean, it doesn't hurt to ask. So I got in contact with Julian. He's like, yeah, so you can come anytime, and then we can get you on track and start for PhD. I'm like, okay,
Wow.
that was that easy. It's very, it's funny. You think you're lucky, but I think most of the time people get to these points, like PhD in search, uh, like that randomly by just knowing someone that knows someone.
Yeah, you never know. While you get a sip of your tea, You were talking about the bacteria and the microbiology and everything. So it reminded, and it's something I don't think about that often, but it reminded me of the fact that and I did my high school in Canada, actually just did 2 years and finished high school there. And in Quebec, before you go to university, there is what they call cégep. So it's 2 years of extra prep before you go to university. And during that cégep, I took a microbiology class, and actually, we did exactly what you just described, where we had to collect samples across campus and then grow the bacteria on different plate types. And then we had to characterize the bacteria. And it was so much fun. It was a lot
And
of fun.
and we
Um,
were also I remember because with this class we were actually in charge of testing stuff sometimes throughout the
okay.
school. It was a boarding school. I didn't mention that.
Okay.
It was a boarding school. It looks like awkward.
Hogwarts, you mean the
Awkward.
Harry Potter?
Yeah, it was a castle, an old ministry, and they
Wow.
converted it in the high school. It was very fun. I really had a great time over there. And we were in charge of testing in our microbiology class, water, the restroom. They asked us to swipe stuff within
Oh
the high
goodness,
school.
that's scary whenever it grows.
Yeah, it was not great. I mean,
Yeah,
let's put it that way. We had plenty of material to work
no, that's for sure. And it reminds me of the fact that, these places are not necessarily clean and there's a lot of stuff growing on there. Yet our immune system, how beautiful is our immune system?
with. Mm,
Because I don't want to say, well, maybe not everybody washes their hands properly, or most people don't wash their hands properly. Which reminds me, I was volunteering at Saint Justine Hospital. This was during university, and we were doing an exercise where you got this UV dye on your hands, and then you had to go wash your hands with soap. And
mm.
then they came with the UV light to show where are the spots that you missed. And we were 20 people in the class, and none of us got it right. None of us. And then you could tell where you actually didn't scrub the thing off.
And
So
like,
it's a miracle. The immune system is really phenomenal with everything growing everywhere.
we are lucky, that's for sure.
Yeah, yeah. All right, so you're moving from France to
Yeah.
Iowa.
So we did that within the first, I mean, we talked for like a week or two, and then within a month or two, I was in the US. So I quit my master, which was actually very stressful because we switched. It was during the period where the new school year was starting, so I was really leaving at that point. I was, if I weren't leaving, I would be in a deep trouble because I was not even registered for M2. So it was either I was going or I was taking a year off.
Okay.
So I was
Wow.
very happy to be able to ended up going, and it was like my first big project in my life where I had to take a big decision like that
Yeah. Yeah.
without my parents.
Of course. Can you tell us a little bit more? Just because I don't know it well enough the French system. How does you said master one and then master two. What is the difference between the different master or what's it called? The different degrees. So you said that you were not registered. You
Yeah,
would have
so
had to take a year off and then you quit your master. Does it mean that you didn't get your master's degree and you went for a PhD, or what does that look like?
So no, yeah, I never got the final master. I just got my pre-master, master 1 year, basically. So
Okay.
it was great for master one. Probably not valid anymore since it's been nine
Okay.
years, 8 years.
Okay.
Probably not valid anymore. So I would have to start from the beginning. But yes, you do 2 year of master, and then you graduate with a master in
Okay, so
like
you only did one.
usually. Yes, I did one
Okay.
year.
Okay. I
Yeah,
was confused there. I thought it was like 2 degrees or something, or what did
no, no,
that look like? Okay. But then now you have a PhD, so who kind of cares at this
exactly.
point?
Exactly. My master, I want to say the master I was into was excellent. It was a translational physiology translational. I don't know if it translates well. Translational physiology.
Yeah, I
Uh,
think it's translational physiology. Yeah.
it was awesome, and the teacher were great too. I mean, I didn't leave because I didn't like people. I
Yeah, yeah,
really
yeah.
I had the opportunity. I couldn't go.
Yeah. And then what was your PhD about?
So, I started, so the first 2 year of my arriving to the US. So I arrived to the US, and the first 2 year were me trying to get my F1 student visa,
Mhm.
and because I came on a J1, uh, and I needed a F1 to start the PhD program, and I was going through direct entry. I was supposed to take this, I forgot the exam name, it's GRE.
Yes, I think maybe GRE. I again did studied in Canada, so I don't know.
So there is an exam that you have to take to get into a PhD program. I think it's GRE, and
Okay.
it's a combination of language and math and stuff like that. It would have been horrible if I had to take it because I don't test well, especially when language is involved.
Yeah.
So, science is fine, math is okay, language not at all. That's why I went into science. Um, but they canceled it right when I applied for the program. So I didn't have to
Okay,
take
good.
it. I
Good.
was very happy about it. So the 2 years, I spent my 2 years first reading a lot of literature to understand what I was doing there. Julian trained me on many, many technique, molecular genetics, uh, some in vivo a little bit. And then I joined the program, and the program was molecular physiology and biophysics
Mm,
and the University of
interesting.
Iowa. And then I did my PhD. I completed my PhD within 4 years. And during that time, I have studied not a GPCR per se, but an accessory protein called the melanocortin receptor accessory protein, or MRAP2.
Okay.
Uh, there's an MRAP1 and there's an MRAP2. MRAP1 doesn't do much, and MRAP2 does a lot, so we focused on MRAP2. And it regulates many, many, many GPCR to the point where we were discussing with Julian about renaming it, keeping the word MRAP2, but renaming the letters and naming it like master regulator of GPCR or something like that. Because every time we were testing something, it seems to have an action or an effect on the
Mhm.
GPCR we were testing, and it ended up not being the case. But the one we tested were so of course MC4. It was discovered by Roger when he cloned MC4, and they found along with MC4 MRAP2, and it's an accessory protein that it's actually very funny because it's one of the only protein that is known. I don't know if it's true still now, but back when I was studying it in like 2, 3 years ago, it was known to be the only mammalian protein that is able to form an anti-parallel homodimer. So it means like you have the one C-terminal on the outside and one C-terminal on the inside, and they bind together like this
Wow.
unit, and they bind to a receptor that they regulate. And the one I studied the most, I started with the orexin receptor, and then I moved on to the ghrelin receptor, GHSR1A,
Hmm.
which GHSR1A incidentally might be my favorite GPCR,
Yeah, yeah.
with close second MC4, but we're still, getting to know each other. Um,
Yeah.
But yeah, so MRAP2 we shown that MRAP2 has massive effect on those receptor. GHSR1A is known to be very unstable when it's not bound to anything. It's always on off, and it has a very high constitutive activity. So when you look on graph, you see that it looks like it has an IP3 effect that is very high,
Yeah.
all the time. And when MRAP2 binds to it, it shuts down this constitutive activity so that you can see that when you stimulate the receptor, the maximum response is the same, but the amplitude of the response is much different. So it
Hmm.
looks like a potentiation of the receptor signaling. Um,
Interesting.
and it also helps trafficking some of GPCR. It helps them traffic the membrane. MC4 is a good example. It also, and it doesn't only potentiates receptors, it also inhibits the activity of other receptor. So it's kind of a modulatory protein that is expressed. So we don't know if it's dynamically expressed in the cells. I don't know if it's a constant expression or if it can change based on, I don't know, on food intake or energy status. Um, we don't know that, but it could be a way for the cell to regulate the the signaling of its receptor. If it needs to be more GHSR1A, it's going to increase the amount of MRAP2. So it's going to activate GHSR1A, inhibit the orexin receptor.
Of
I
course,
don't,
then.
but yeah, it's pretty cool. That's why it's my favorite access to the protein so far.
Quick question. So I just want to go back a little bit. So you're moving to Iowa. What does
Mhm.
it look like for a French guy to move to Iowa? I mean, the reason I'm asking is because it's first of all, you're changing countries for the first time. What
Right.
a big contrast between France, Europe, and the US. And then it's Iowa, which is again a very different environment. So, typically people when they move to the US, they either stick to the East Coast or the West Coast. And I guess
Mhm.
you followed the science, you followed the scientists where you wanted to do your PhD, but what was it like for you?
Yeah, I mean, overall, I'm a small town boy, so I don't really enjoy big cities at all. So I'm more comfortable in those kind of city. Iowa, so in Iowa, the city was Iowa City. It's a
Mhm.
small, progressive city. It's totally my kind of city. Actually, I would love to go back there. Um, it was weird, especially that my English was not great. It was okay. English is my third language, so I had to relearn, how to say, I had to learn how to think in English, uh, and it was an abrupt uh, curve. It was not very like smooth. It was,
Yeah, you were a steep learning curve. You were dropped
yeah,
in the middle of the pool and go swim, basically.
yeah. So the headaches. I don't know if you experienced that. When did you were you always English speaker or did you learn to speak
No, no, I learned English, but for me, I think it was more like a switch than anything else. So I'm half Algerian, half Hungarian. So my mother tongue is Hungarian, but I learned Arabic at school. I went to school without knowing the language at all. It was a very scary day, but I used to speak French as well. And then we moved and lived in Canada where I studied in French.
English?
And then during my master's or PhD, beginning of PhD, master's of PhD, I had the opportunity to go to a Gordon conference in Italy. And I think this was 2005, maybe. I'm just trying to rethink. And I shared the room with a PhD student who was at McGill University in Thierry Hebert's lab, but she was from Italy, so she didn't speak French. And this was my first international conference, and I was presenting a poster. And I think at that week, my English just turned on. But
Mm.
before that, I barely used English other than to read papers because my classes, everything was in French. My homework was in French. My master's degree was written in French. My PhD degree was also written in French. So yes, it could have been this thick, but because it was in French, it was about this thick. Because you need 3 or 4 times the number of words to express the same thing as you would in English.
Exactly.
Um,
I'm a terrible French writer. Terrible.
it's hard. It's very difficult.
It
It is
is. Oh,
extremely difficult. It always reminds me, there's la grande dictée.
mhm.
Um,
Oh, yeah,
that
yeah.
one is like the next level. So for the audience listening, I think it's a yearly contest where there is somebody who's reading a text in French, and basically you have to write down what you're hearing, and then they check for spelling. And those are the most complicated words in French that end up in that text. Um, just
Yeah.
listening to it is just mind blowing.
Just the thought of it is
Yeah.
hard to, yeah. No.
Yeah.
Thank
Yeah.
you.
But it was a long-winded answer, but it just turned on for me. And
Okay, so you didn't get the, because me, I know that when I transitioned, at first I was translating everything in my head, and it was
mhm,
first it was time consuming, but also it was exhausting. I
yeah,
remember like first month in the US, I was in bed at 9 p.m. Like, it was insane how much energy it took me to, and then it gradually changed, and I started to just think in English rather than translate my
yeah,
thoughts. And then from there on, it was much easier. But it was a rough adaptation. And I see that I have a friend that came to do his postdoc while I was in my PhD. He came to do his postdoc at the University of Iowa as well. Paul Buscaglia. He he had a lesser, I would say that his curve was smoother, but not in a good way, I think, because we were talking in French. I think I was slowing his progress
ah,
in English, and as soon as I left, he blew up.
yeah,
Like he had like
yeah,
great English, and he started to
yeah,
speak much better. So I think
yeah.
I slowed him down, and I'm sorry about it.
And it's also on record. No, it's interesting because people often ask me this question, in what language do you think? And I can't answer. I don't know. It depends. It depends. I have a hard time. Well, I have an easy time to switch from one language to another. If it's a language that I use regularly, I don't know what language I think in. I think I don't even I can't figure it out. It's like there is some thoughts that happen in my head, and then my brain picks which language it activates when that
Mm.
happens.
Yeah, I do that too, based on emotions. I'm usually scientifically, I always think in English,
Yeah,
but it's more like a social uh,
yeah.
setting. And I often catch myself thinking in French.
Mhm.
But yeah,
Interesting.
it was a rough transition.
So you mentioned also, I wanted to talk a little bit about this because the reason why I brought this up is to ask about how your transition was from, one country to another, and also a different language, is because most of our audience, or a lot of people listening to the podcast, are between the ages of 25 and 35. And I always want to hear how the transition went for our guests in order to encourage them. Because although it was difficult and your learning curve was steep to get to the English that you want, level that you wanted to, and push back your um, going to bedtime because of the energy expenditure, I think it seems like it was absolutely worth doing that transition, going
Oh
from
yeah,
from
I do
Europe to
not
the
regret it at all. Oh yeah, yeah, for sure. No regret whatsoever. As soon as I came to the US, I was very well supported with Julian, Dr. Seabag. He's also an immigrant. He came from France as well. Um, and he really like fought for me. He took care of all my J1 F1 transition. And when there was a problem, he was up in arm and ready to defend me every time. So I really never felt the immigration problem until I actually left his lab and had to, sponsor myself. And this is where I realized it's like, oh my God, it's so much work. But
yeah.
he did great. He really helped me a lot, along with the University of Iowa, in transitioning and maintaining my visa.
Yeah, yeah.
So you just have to find the right people. Yeah.
Two more questions before we move on to your current position and the transition from Iowa to Michigan. You mentioned something about three languages. So we have French, English, and what else?
And German. That was my
German.
second
Wow.
language.
Wow. That's hard. That's a hard one. At least for me, I had German classes in school before I moved to Canada, and that was just absolutely zero. No permeability at all. So wow, kudos to you. Do you ever think in German?
No, never. I never lived in Germany or I've been immersed in the culture, so I don't think my German spoken would be even great. If I were to speak with someone, I don't think they would find it okay. I have
Would
never
they, would you understand what they were? You couldn't be sold, right, in German.
Yeah, no, probably not.
Okay. All right, and then the other question I had is, um, you mentioned having great support from your PhD mentor from Julian, and you really had a good time. When you decided to move to Iowa to his lab, you mentioned you were in discussions for about a week or two before everything happened. What
Mhm.
gave you the confidence, or what were the signs that you got from him that gave you the motivation to say, okay, I think it's going to be okay, and I really want to do this? This is important because you're one of the exceptions who didn't write to many or email many different people to figure out which lab they wanted to go to. It kind of happened through a conversation. But I want to know, what were the criteria, even unwritten ones, that pushed you to say, well, actually, this is going to be cool. I'm going to go there.
Yeah, I should say that I actually applied to a couple
Oh,
uh
that's
PhD,
sorry.
but so I didn't know how it worked. So I was just emailing PIs and they were like, sorry, we're part of an umbrella, we can't take you. And I'm like, what is an umbrella? What are you talking about? And they were not once they rejected you, they intended not to talk to you anymore. So I didn't understand. But yeah, so Julian is a very confident individual and is very well spoken and can sell you anything. And he sold his lab to me very quickly. He just basically described what he was doing, what I will achieve, and he didn't lie. I did great in his lab. The first year I came around, we published one review, and basically it was a review just recapping what MRAP2 is used in MRAP1 as well. What
Mm-hmm.
does it do physiologically?
Interesting.
And then what was I doing? What did I do? I was doing something with neuronal. What was it? Oh yeah, I was trying to understand whether MRAP2 had an effect on neurons, and we had the idea that we weren't sure, but we had clue that suggested that MRAP2 was also expressed in mitochondrial membranes. And we did see horse experiments on primary neurons. And apparently, for some reason, it was never done. So on arcuate nucleus, well, no, yeah, they were arcuate HGRP and PY neurons. And apparently, it was never done. I was doing microdissection, dissociation of the neuron, and then put them in the seahorse. Uh, and then I was isolating. Sorry, I was isolating mitochondria and putting them in the seahorse. And in collaboration with another lab, we wrote a book chapter on the method we developed to check the oxygen, the activity of those mitochondria. And then the third year, I published my first author paper where I described the effect of MRAP2 on orexin receptor and PKR1. And then we then moved on to GHSR1A, and I published again as first author a paper on the effect of MRAP2 on the GHSR1A, and then another paper when I was writing my thesis on the effect on how MRAP2 regulates GHSR1A. We already knew the effect, and I did some a molecular experiment to determine how MRAP2 prevents the constitutive activity of the receptor and how it help in recruiting beta arrestin. And and what was your question again? Sorry.
No, so the question was, actually, I forgot my question. I got to thinking about MRAP2 because I honestly don't know enough about it. So, and the other question, the other thing, one of the questions that I have listed is, how did you get into studying GPCRs? But I guess working on MRAP2 got you into hearing and working on GPCRs, although your focus one was on MRAP2. So I'm very interested in a high level description as to, you mentioned it a little bit, but a little bit more details about what does MRAP2 do, any historical facts. I don't want to put you on the spot. If you say, Yamina, here is my review, read it, that's fine too.
No, so MRAP2, the problem with MRAP2 is that physiologically MRAP2 knockouts become, they have a late onset obesity. So it's an accessory protein of MC4, and MC4 is well known to regulate food intake and energy homeostasis. If you do an MRAP2 knockout animal, they have an early onset obesity and they have a hyperphagia, decrease activity. MRAP2 is kind of more tricky because it doesn't look like there is any phenotypical response that would suggest obesity, but yet there is an obesity phenotype. So they have normal food intake, they don't have decreased activity. We don't really know how it works. Apparently, it's how MRAP2 distribute the fat is altered in some way. And I don't think we have the response yet, the answer to that yet.
Interesting.
But
Sounds like a no, no, please go.
but
No, I was just gonna say sounds like a very interesting protein. There's a little delay, so it looks like we're interrupting each other.
no, it is an awesome protein, and honestly, this is I hope I still like I shouldn't say that, but so basically every time I do an experiment, I always try to include, because MC4 is regulated with MRAP2, and I study a lot of MC4 right now. I
Yeah.
always try to have a little condition with MRAP2, with or without MRAP2, to just continue a little bit, keep the legend alive.
Yeah, yeah, you never know. I mean, there's so much that we don't know, and you don't know what you don't know. So if you
Mm,
have space on your plate or space in the experiment where you can add in an extra condition.
exactly, it doesn't hurt anybody,
Yeah.
and my favorite discovery with MRAP2 was right before I left, and I showed that MRAP2, let me say, oh, sorry, I'm gonna reformulate that. It's not my favorite discovery about MRAP2. My favorite discovery in the context of studying MRAP2 in GPCR-1A was that GPCR-1A appears to have a lack of phosphorylation when MRAP2 is bound to it. So it prevents the better recruitment of beta-arrestin. And the interesting thing is that when you so MRAP2, since MRAP2 inhibit the beta-arrestin recruitment of GHSR1A, we wonder why and how. And I showed that when GHSR1A C-terminal tail is phosphorylated, it actually doesn't serve as an anchoring point of beta-arrestin, which is not the current model. There is multiple model, but the most accepted model is that beta-arrestin use the anchoring aspect of the C-terminal tail to get into the core of the protein. And I showed that by doing just a simple deletion of the entire C-terminal tail of the receptor, I showed that it actually increases the recruitment of beta-arrestin. And I developed what I call the gate model. So the C-terminal tail is actually a gate model. So it just sits on the core of the receptor and prevents any recruitment from beta-arrestin. And I was never able to prove it, but my hypothesis is that when it's phosphorylated, this C-terminal tails moves away, and then
You have
beta-arrestin access to the core. And the same theory was developed by another lab. I forgot the name of the person. I'm terrible with his name. He works at the Mayo Clinic, and he showed the exact same thing with a different receptor. That
interesting.
like the C-terminal tail was used as a gate model, a gate rather than an anchoring point to beta-arrestin. And
I love it.
it was during the GPCR retreat in Niagara Falls.
Hmm.
He presented these results.
Oh, interesting. Now I feel like I want to go back and see the program and see who this is. And in
We emailed back and forth after. I could
okay,
give you,
okay. I'm
but it was very
curious.
validating that someone else find the same thing that I did,
Nice.
in
That
time
is
period.
really nice. It's very interesting. And it's funny because whenever you think, well, when I in general, we think about GPCR as an activation, we do, the classical assay, G protein, beta arrestin, but then there's so much more that we don't know
Yeah.
that happens in the cell. And there's so much that controls receptor activity
Mhm.
that we either don't know about or don't yet have the tools to study or to measure.
Right. And this is basically from there on, I actually developed a massive interest for bias signaling. And this is if I continue in academia, it's probably what I want to study, stay on to be able to. It blows my mind that we screen entire library of compounds for just protein G activity, and we just bypass beta arrestin. When if you find a low or no or G protein response, but a high beta arrestin response, it could be invaluable because they also have their own signaling.
I agree. I agree. And yeah, I absolutely agree. I we invited to the Dr. GPCR symposium, not the one in November, but the one in September, Terry Kinakin. And I love how he thinks about receptors in general as these allosteric machines
Hmm.
where anything can be an allosteric modulator, whether it's a G protein, whether it's the ligand, or everything is in motion. And so you everything, any interaction causes an effect. So
Right.
kind of removing the labels of G protein, beta arrestin, anything else. If you look at the system and say, well, this is my protein of interest, which is the receptor, and I want to look at what does modulate its function and where does that come from. And then it's hard to integrate, but it's still fascinating to think that different cells have different proteins and they still able to integrate the signaling and do the right thing at the right time, knowing that there's not just one GPCR per cell, there's many more.
Hmm.
And
And there's
it's just,
you also have those GPCR that can do heterodimerization with other GPCR. So
yeah,
all of it is dynamic, and based
yeah,
on the interactome of the cell, you're gonna
yeah,
have different response for the same agonist and the same
yeah.
receptor.
I think it's fantastic. I think it's really, really cool.
Agreed.
All right. So how did you end up in the lab that you're in today?
So Roger Cohn was the PI. Julian did his postdoc in Roger Cohn's lab.
Mhm.
So we collaborated a lot on different projects. I published my second paper. I published with two of his lab member,
Okay.
and then we had multiple visits over there at the Life Science Institute where I'm at now. Uh, and at the end, I didn't know a lot. That's one of the things that I lacked during my PhD, and I would recommend for the PhD student now to actually invest a lot of time in networking and meeting people because I didn't know much anybody else than Roger, my committee meeting at the University of Iowa, and Julian,
Yeah.
basically.
Yeah.
So I went for, and Roger is an excellent researcher. I didn't settle down. I settled up. I was very lucky that he accepted me to be in his lab, honestly. And I also wanted to stay in the same area of, back in the day, I really wanted to stay in MRAP2 environment.
Yeah.
Even though we don't do a lot of MRAP2 study in Roger Cohn's lab, but he's still interested in MRAP2, but it's not his main focus.
Mhm.
So this is how I got there. I basically asked Roger if I could join his lab, and since he knew me and I knew him, it was very easy.
Yeah, which is again goes back to what you were mentioning in the beginning is you never know where you're going to end up. So keeping an open mind, talking to people, and then especially if you're open to collaborations, you go to meetings, you get to meet people, then it's so much easier to prepare for your next step.
That is very true. And during the meet, that's the thing. When you go to a meeting, I used to just go to the talk and then move on, go do something, or because we were with friend, we didn't talk to anybody else. This
Yeah.
is really the wrong thing to do. You have to engage with people. I know it's uncomfortable at first, but you get used to it very quickly because it's very rewarding at the end of the day. It's scary at first, but then when you do the first move and you actually talk to the person, you realize you're not dying, and then you actually enjoy out of it, and it's gonna just get easier to do it. But starting that early on is a very good idea.
I love how you, you described like you're not dying, and it is true, but it can be as daunting as you're like, oh my God, I'm gonna die. I have to talk to that person or to a one person, and which it's so interesting. It's no, it's like confidence. You are not born networker. You are not born confident. You build, you gain it over time. So, for those listening who have never networked or never gone out of their comfort zone, you can start step by step, small little steps. And then that at some point, within 6 months to a year, you're going to be a different person
Yeah,
because you're growing your ability to network.
start by talking to a PhD another PhD will like that anyway. So find a PhD that is just like you, not talking to anybody, and just
Yeah,
go talk to them, and they're gonna
yeah,
introduce you to their PI and stuff like that, and just
exactly. Exactly.
and
No, I think, oh, please go ahead. Again.
and the more the more people you know, the better choice you have at the end of the day.
Yeah.
The more people you can choose from. I don't know how to say that correctly, but you
Yeah,
have
no,
more
I agree.
of choice.
Yeah, yeah, yeah. You have more opportunity. And I think it's important to know what others are working on, what others are doing. And it's so easy. You can kind of even build yourself a protocol on how to approach someone and
Mm.
what are the questions that you could be asking that person. The
Yeah.
more you ask that person about them, what do they do, where are they from, what do they work on, and the more you make them talk about themselves, it sounds silly, but the more they're gonna like you because you gave them the opportunity to talk about themselves, and then you're
No,
building that bond.
I remember having those very awkward moments at first when I was trying to interact with people. Oh my God, that was horrible. They were blank and nobody tried. So always have, a fill the blank remark or question that you can ask the
Question,
person.
yeah, yeah,
Yeah, have 10 of them, and then you're gonna fill all the blanks, and nobody's gonna be
yeah,
uncomfortable.
exactly. And it depends on people. Sometimes, silence is not a bad thing. It's very uncomfortable, but once you've chatted with someone a little bit and then there's some quiet moments, obviously you're not standing in front of each other, but maybe you're at a conference and then you just chatted a little bit and then there is a moment of silence. It's not a bad thing. I
No, no,
think it shows that you're comfortable enough with that person to stay quiet
right,
for a few moments. But yeah, the weather is a good, especially if you're in Canada or in Michigan. Talking
exactly.
about the weather, there's a small little topics that people might be interested in. The other thing is that getting prepared. You're going to a conference. Yes, typically as a student or a postdoc, you have a poster or a talk to give, right? So you're preparing scientifically, but there is an additional set of preparations that you can do. You can look at the list of speakers. If you have access to the list of attendees or people who are going there, then you can look up those people and see what they're interested in, read a paper or an abstract from those who are presenting or poster presenters, and this way you can go and have a productive conversation and you can say something nice or you can ask a question about previous work that they've done, and again going back to you asking them questions about what they're doing and who they are and what their work is on, and it's that simple.
Yeah, the first step is always the hardest, but
Yeah. Yeah,
take.
but that's a long game. That's a long game. You never know, who you will end up working with, who you can help, who can help you, but it's all about forming those connections.
Yeah, no, for sure. I mean, I would have never gone to my PhD if I hadn't talked to that person that I didn't know back in the day, but
Yeah, yeah.
social,
At the end of the stairs.
yeah, the cloud of cancer.
Yes, yes. It reminds me of this episode of Friends. I don't know if you've ever watched Friends, where
Yeah,
when Rachel got this job, I think at Bloomingdale's or something, and then the boss and the colleagues are smokers, and they make all the decisions without her while in the smoking area, and then she tries to act like she's smoking just because she wants to be in the conversation.
It is so true though. My best interaction with the PI I was doing my internship with it was when we were smoking downstairs.
Yeah, because you're in a different setting. And it was a
Yeah,
different time as well.
it was a different time.
All right. So yeah, now you moved to Michigan. You're in Roger Cohen's lab. You have this interest in the melanocortin receptors because of your PhD work. What are you working on currently, if you can tell us?
Yeah, so I'm working on a lot of things. I
Well,
started.
then once you tell us how many things you're working on, I'd like to know how do you manage all those things?
Uh, do I?
I don't know, maybe.
I mean, I definitely do, but I would recommend not to have too many things. Cut back. So today I actually had a meeting with my PI Roger, and because we talk about doing something new, and I'm like, okay, we do something new, but now we have to cut somewhere else because I can't. Um, so I do a lot of things. So my main focus is on the melanocortin 4 receptor. So Roger likes to assign specific project to his postdocs, and then you can also have additional project on the side that it doesn't, you know, he lets you do whatever you want, be creative,
Mhm,
um, which is awesome. So since I love biosignaling, I have been working with a compound library that he has developed in collaboration with Courage Therapeutics. It's a full library of agonists, MC4 agonists, and some of them
mhm.
were extremely specific for the first time and to the MC4. Oh yeah, there is multiple MC4. There is MC1, 2, 3, 4, and 5, and they lack a lot of specific. I don't know if you heard of setmelanotide.
Well,
It's
all of these things are always in the news lately.
Yeah.
So,
So setmelanotide is a MC4 agonist.
mhm,
It's not specific at all. That's the problem. So if you are going to inject, it's an injectable drug. So if you inject it in your skin is going to darken because it's going to activate something else. It's going to activate another melanocortin rather than the MC4 that
mhm,
is expressed in the brain. And when you activate MC4 in the brain, you are shutting down appetite. So basically,
yeah,
you are increasing satiety. Um, so the idea, it works, but it's not specific at all. So what we are trying to do is develop specific MC4 agonists.
okay.
Uh, and from this library, so they developed a ton of compounds through this effort. And through this library, I actually tried to screen it for also beta-arrestin. So I had to adapt an assay that we developed when I was in June Sibak's lab. It's a nanobit-based assay.
Mhm.
Are you familiar with nanobit?
Yes, yeah.
Well, I'm glad. Maybe other people are not. Sorry, I forgot we're not
But
just two.
yeah, yeah.
So nanobit is a split luciferase. There is a large bit portion and a small bit portion. And when they come together, they reform an enzyme, a luciferase, that is able to hydrolyze the substrate that you give it. The substrate is used, I think, is called furimazine. So through that, you can have MC4R linked to fuse the C-terminal of MC4R fused to large bit, and you fuse the small bit on the N-terminal of beta-arrestin. 1 and 2. And then when you activate the receptor, it's going to be phosphorylated and recruit beta-arrestin. And when they are approximately close to each other, they are able to reconstitute the enzyme. The enzyme is not, the affinity of large bit and small bit is extremely poor. So there is no they don't drive the interaction between the receptor and the beta-arrestin. So only the spatial parameters is the only one that matters. So when they're together, you have reconstitution of the enzyme and you have the luminescence when you hydrolyze the substrate. Uh, so we developed this assay. I had to just adapt it for high throughput screening,
Hmm.
which was very easy since the assay is excellent. Um, and then I started to screen the entire library, and I found a couple nuggets that indicate that there may be some beta-arrestin agonists that are biased towards beta-arrestin. Sorry,
Okay.
I'm trying to point that phrase beta biased. It's not
Mm.
working so far. I didn't publish it, but I will if I can. I'm trying to call beta biased agonist, so an alpha biased agonist, so beta-arrestin
Yeah.
biased
I I
agonist.
think
So
I think Lawrence Slosky has this it's well actually it was a modulator, but it's beta arrestin the BAM, the beta arrestin allosteric modulator. I think that's the compound she's working on. So maybe a cool acronym uh out of there.
yeah, I mean, I'm just trying to, because every time I have to explain that, oh yeah, this is a biased agonist, it's biased
Yeah.
towards beta-arrestin, whether I can just say it's a beta biased agonist.
Yeah. And in this case, you mentioned what are you looking for exactly?
So I'm trying to look for in uh, so beta-arrestin signaling is very poorly studied.
Hmm.
There is very little data on the bias signaling of beta-arrestin signaling, and the in vivo effect of beta-arrestin activation is very poorly studied. So what I'm trying to do is to find a beta-arrestin biased, an agonist biased towards beta-arrestin, so a beta biased agonist that I can
Hmm.
inject in animals to see what the activation of only beta-arrestin through the MC4R receptor, what would
Okay.
be the effect? Do we get satiety or do we get cardiovascular events? Beta MC4R is known also to have, when overactivated, to create severe depression, and you will have a lot of there is two examples. There is a hypo and hypertension events that happens.
Okay.
And that, as you may know, in drug discovery, it's a big no-no. So as soon as you get an effect on the heart, they shut it down. So by it, the idea of being able to decouple the signaling of the receptor and isolate those effects. So let's
Hmm.
say beta-arrestin signaling is mainly giving a cardiovascular effect, and GS is mainly giving a satiety effect. So we can develop, better drugs. So create an
Yeah.
alpha biased agonist to make sure that this guy is only going to trigger the satiety effect and not the cardiovascular effect. So that's
Yeah.
what I'm trying to do, but
Hmm.
it takes time.
And is it beta arrestin 1 or beta arrestin 2? The reason I'm asking this is because for every GPCR I worked on, I always tested beta arrestin 1 versus beta arrestin 2, and very rarely I saw any difference between these two. Although we know that beta arrestin 1 is also involved in the beta catenin signaling pathway. But
Yeah,
any
so
thoughts on?
they do both. So MC4R recruit both. A preferentially recruits beta-arrestin 2, but again, this is an overexpression system I have been studying it with, so I couldn't tell you if it's true or not. But
Mm-hmm.
from the in vitro the study I did, it looks like it's preferentially recruiting beta-arrestin 2.
Interesting. Interesting. That's why I keep wondering why I always tested. So I used to use Brett to measure beta arrestin recruitment. And no matter which beta arrestin I looked at, any receptor, mostly class A, mostly chemokine receptors, no difference between beta arrestin 1 or 2. We always stuck to 2 if we had to make a choice and decrease the number of plates we had to run. But I in these in vitro assays, I never could find a receptor that had a preference for one versus the other one.
Yeah, oh yeah, I don't know if it's like I said, I can't tell you if it's just a expression problem. I didn't check yet.
Yeah,
It could very well be just that. And I've
yeah,
never encountered I never encounter a GPCR that doesn't recruit either, both basically.
yeah,
If you shut down beta-arrestin 2, it's just going to recruit more beta-arrestin.
exactly. Yeah, yeah. So there's this. Maybe that's the whole point of having two beta
Yeah, they
arrestins.
compensate each other.
Yeah,
But yeah, I don't know if you saw this assay. It's superior to the bread because you can see kinetically the effect, the recruitment of beta-arrestin. Uh, so let me actually, well, no.
now you can if yeah, but I was gonna ask how with Brett as well, you can very nicely see the kinetics of beta arrestin recruitment. Depending on which generation of breath biosensors you have, you can measure at room temperature up to 45 minutes easily. I don't know
Mm.
about the nanobit, but it's very nice. And then you can actually, at least what I've done in the past with chemokine receptors is either you have the cells plated, you add your substrate, and then you add your ligand, and then you put in the plate reader, and then you measure over 30 to 45 minutes, and you can very nicely see the recruitment, or the opposite, where I was looking at a decrease. So I
Mhm.
stimulated the cells for 5 minutes at 37 with the compounds, then get the plate out, added the substrate, and then I was looking at any decrease.
Right.
Um,
Yeah, it's relatively similar. You get like very the definition of the signal is um, superior because you can use PMTs with luminescence, right? So
yeah.
you have a better definition of the kinetic. So you're gonna see the recruitment of the beta arrestin to the GPCR, and then you're gonna see the decrease, and you're gonna see dissociation between
Yeah.
GPCR and beta arrestin. And some of them have, recruit and steady, so you can tell they are
Yes.
heavily bound together and they're very high affinity with each other.
Yeah, that's what I was gonna say. I don't think I ever saw like really quick dissociation.
Oh, GSS1 does not like beta arrestin 2. As soon as you can get rid of it, it does. It takes
Interesting.
roughly within 2 minutes, it just kicks it off.
Wow.
But
Interesting.
it tells you whether or not the receptor is either internalized or recycled, or it gives you clues like that that you can exploit.
Exactly, exactly. And then you have the bystander Brett tools as well that allow you to track your receptor and see where it goes after stimulation. So it's really cool. So that's one project you're working on. You were mentioning working on multiple projects.
Yes. So I'm also trying to. I always wanted to do that, never was able to do it. And since Roger Cohn has a little bit more money than Julian, I wanted to test and determine and finally assess a mosaic, a phosphomosaic of a
Okay.
GPCR. So I want to stimulate it. I'm working on stimulating MC4R and trying to catch that frame and send to mass spec and have to identify the phosphomosaic of MC4R
Mm-hmm.
based on the agonist I used. So the idea
Okay.
is that if you have a balanced agonist, you're gonna have, I don't know, like 6 different phosphorylation sites that are going to be phosphorylated. And if you use a beta arrestin agonist, you're gonna, I don't know, maybe 4 of them are going to be phosphorylated. So you can identify important phosphosites that are used for this and that.
Mm-hmm.
So this is one of the things I'm trying to do right now. It's just super hard to get enough material. Uh, I'm trying to do that in plates. It didn't work because the maximum plate we have is a 15 centimeter, and
Mm-hmm.
I tried with 10, and I didn't get enough protein to be able to see it on the Kumasi. Because if you don't
Wow.
see it on Kumasi, don't go to mass spec. It's useless.
Yeah.
And especially I mean, we tried with a silver stain. We can see it with a silver stain, but the thing is, it impacts then the cutting of the protein later on when they have to before they run into mass spec.
Okay.
So uh, so we have a great structural biologist in our lab called Alice Persley, and she is used to do it producing a lot of protein to be able to see them through cryo-EM. Oh yeah, we have a cryo-EM at the Life Science Institute. So
Nice.
Alice is comfortable there. She can do cryo-EM all the time and do crystal structure and stuff. She is very lucky for that. So she knows how to do these kind of things. So she helps me, and she helped me in finding a way to produce enough receptor that I will be able to stimulate and and shut down stimulation in the frame I want. The hard part is to find the right time to stop the reaction to keep the phosphorylation because they can be quickly dephosphorylated right after.
This is
And it
super
has
interesting.
Yeah, and it has to be done in mammalian cells. This is the problem. It could be very easily done in yeast or insect cells, but in insect cells, you're gonna have maybe you're not gonna have the right GRKs. You're not gonna have the right
Yeah.
PKAs,
Yeah.
and MC4 apparently use all of them. So I have to find a cell line that have all the good kinase that can phosphorylate my receptor correctly. So yeah,
Any
we're gonna try to use that.
any chance to do this in a cell-free assay? I don't know.
That's the, you need to supplement it, right? So
Yeah. It's
I
more
tried.
work, but it might be cleaner.
The thing is, I don't know what kinases I need to add. It looks like you use most of them. I did the inhibitory assays where I use compound 101 or
Yes.
Geo 1693 that blocks GRKs 2, 3, and some of the
Hmm.
PKs. And every time I block, there is still phosphorylation or still beta arrestin recruitment suggesting
Yeah.
phosphorylation.
Yeah.
So I haven't been able to identify a specific kinase that I would have to supplement in a cell-free assay.
Yeah. Have you thought about looking at like GRK knockout cells or even beta arrestin knockout cells where you would supplement, a kind of a cleaner background where you can add in the proteins that you want and see the different effects and then from there, because for example, I worked with compound 101. I mean, it's such a messy thing and you need to use a lot of it and it's not that specific. So
Yeah.
it gives you some idea, but it's not perfect. So I'm
Mhm.
wondering, instead of using a salt-free environment and trying to, because you need to purify all of these proteins. It's a whole other field
Right.
there. Maybe thinking about using a cellular cell line with a background where you have, or multiple cell lines that have specifically deleted some components.
Mhm.
And then it's kind of like a puzzle. It's a lot of work. I'm not saying it's an easy fix,
But
but
I do love doing CRISPR, so
there you go. There you
uh,
go. And
it could be done, yeah, for sure. But I think we're gonna go with the easy way. And also, you have to remember that we also have to have a cell line that has an endogenous expression of MRAP2 because without MRAP2, MC4 doesn't even get to the membrane. So CHOK1, some CHOK1 cell line don't even express MRAP2 correctly, and some of them express it too much. So we decide to settle on the hex cells. There is a great heck that you can culture in
suspension.
reactors. Yeah,
Oh yeah.
so I think we're gonna go towards that if I do it. Based on our
Yeah.
meeting, it's going to be benched for a while, but
Yeah.
but we will eventually. I will eventually do it at some point when I have a minute. Uh,
Okay.
Another project I'm working on is understanding the haploinsufficiency effect of MC4R. So MC4R, if you have one copy, you're gonna have half the phenotype. So a het animal will be not as obese, but obese
Mhm.
nonetheless. Have basically every half. You're gonna have the activity, have the obesity, and
Okay.
have the hypophagy. So we need to understand how this haploinsufficiency work. Uh, so I'm working on that. I'm also trying to understand compound brain penetration. So we have, like I said, there is some compounds like setmelanotide that are, if you inject a bolus big enough, you're gonna have a cardiovascular event. And what I observed was a massive hypotension. So the animal just stopped moving basically because it has almost no pressure.
Wow.
uh, so, so, but we don't understand why. And the idea is that maybe it's just a brain access problem. So
Hmm.
maybe your compound is just not getting into the right nuclei or is getting into the wrong one and that can be assessed. So we also work on that. And now I am trying to make a sensor that would allow me to measure the agonist recruitment, the agonist stimulation of a receptor. So basically, when the GPCR would be an on conformation, it would give me a signal, and I can assess. That would actually help me do the brain penetration project and see if when I inject a compound, does this part of the brain lights up? It means the compound has access to it. And I forgot
Wow.
where I'm at. There is a couple more.
And how do you, so let's talk a little bit more about how do you manage all of these?
Uh, I have I don't have it with me, but I have an organizer. I think
Yeah,
that's how
yeah.
you.
Like
I
yeah.
I tried to use Google Calendar, but for some reason, at some point I just stopped using it. I forget to put stuff in it, so when I have to write stuff in my
Yeah.
organizer, I always have a physical one,
Okay.
and I
Whatever
have like
works.
detailed timeline.
Okay. I love it. Yeah, I'm a Trello person. And a Google. If it's not in the calendar, it's not happening. And everything I can think of, every little task that I have to do is typically in Trello. Mainly because I need to unload my brain. It helps
Yeah,
me put it down somewhere where I know that I can set deadlines, I can look at it, I can come back to it, I can backtrack it. And it's hard because for a long time I kept things in my head.
but that runs in the background, and that is a little distracting. So as soon as you put it on paper or in a computer, you don't have to think
It's
about it.
out.
Yeah.
Exactly. And it happened many times during my postdoc or PhD where I woke up in the middle of the night to, oh, I forgot this, So now it's in Trello or it's in my calendar or somewhere where I can track it and it has a deadline. And if I don't do it if I forget about it and I don't do it, I get a reminder a day before, which you can set up reminders to any ton, like any number of days before. And
Right.
then you don't have to think about it. It's kind of a really nice unloading of the
That
brain.
for sure. I do the every morning. I just go through the program of the day and go for it.
Wow. Yeah, it's funny because my calendar is very, very packed. And every time I open my calendar recently, I feel my stress level going up because things are moving so much. But I'm getting a handle on that. And I realized not too long ago that I'm more efficient if I have my mornings without meetings, which
Mhm.
gives me enough time to, dig into my emails, make sure that tasks are done. And then in the afternoon, I'm ready for my meetings. So
It's
again,
actually
I
that's
think,
what that's exactly. I do the opposite. I do all my meetings in the morning, and I have no meetings or emails in the afternoon. Try not to.
okay, yeah, yeah, yeah. I don't know. It depends on if you can work later and the time at which you end your day isn't doesn't have to stop at a specific time, which was exactly the case for me during my postdoc and my PhD, where it actually didn't matter because I just needed to finish my things. And I could sometimes finish at 4, sometimes at 6, sometimes at 8, depending on the type of experiments. But with the family, it's a whole other ball game here.
Also, so maybe it's TMI, but after my PhD, I had a massive. I was very depressive. Uh, I just burned out basically.
Mhm.
So from now on, I do not work after 5, or I do not extend my time in the lab after 5. I can
I
work at
love
home,
it.
but it's healthy to put some breaks because otherwise, I mean, science is so easy to just stay in the lab until 10. Like
Yes.
you can find a way. You can find a easy way to. It's easy to find something to do until 10.
Oh, I
Uh,
agree. I
so
agree.
putting boundaries is healthy, especially if you. It's fine when you don't live with somebody, but as soon as you're gonna enter into like
Yeah,
a more personal life, that's not gonna work great. Uh,
absolutely.
in my
No, I
experience.
agree. No, no, I agree with you. And the times I stayed longer in the lab is because I had nothing else to do, and it was just fun to work
And
with my colleagues and them.
yeah, occasionally it's
Yeah.
actually I do it too. Wednesday usually I go to a bar with the colleagues. We have a beer and usually we go back to work and we continue to do some stuff.
Hmm.
So it's fine to do it occasionally, but yeah,
I agree.
not making an habit is better.
No, I agree. I agree. I actually never worked on weekends ever during my postdocs or even my PhD. I not because one, I didn't want to. Two, I didn't want my boss to know because then it becomes something routine. And
Mhm.
then three is because every time I started to plan something for the weekend, it failed. So that was a good incentive to say no. The one thing I used to do during my postdoc, because I used to live 5 minutes door to door from the lab, is start a culture, split cells. On Friday, for example, I used to do a lot of in-plate transfections. So if I had prepared my DNA and my protocol was ready, I could go in on Sunday to transfect so that I can
Yeah.
start my week sooner. But that was it. Spending the whole weekend, no. Spending too much time in the lab, no either. I think it's not beneficial long term. I did, however, come into the lab at 6 in the morning from time to time, where I had to generate data very quickly to respond to reviewers, and that was a unique situation. But I do agree with you to have those boundaries.
Yeah, just not making it the norm is the healthy
Yeah,
way for me at least. I mean,
yeah.
other people are they prefer
It's all
different.
different. Yeah, yeah. All right. So, what next after your postdoc?
Well, so I don't know yet. I've been dabbing into industry right now. So I went to the conference, the American Physiology Society conference, in 2023, where I met Patricia Silvera. Like I said in the newsletter, I don't think she knows half of the influence she had on me. Uh, but she basically showed us how, she explained to us how to get to a position where you have some, not power, but you are, how to progress fast into your, up to where she was, which is the head of a department,
Mhm.
and the right thing to do, the thing that you shouldn't do. And one of the main points she said is like to involve yourself in a, position of leadership at your university,
Yeah,
and network a lot.
yeah,
So at my university, there was at the University of Michigan, there was we have the postdoctoral association, and they have election every year where you can apply to become whatever chair is open. And I
yeah.
became the chair of international affairs at the University of Michigan Postdoctoral Association. Uh, and so I started by just helping international students, postdocs that, come to the US for the first time and have no idea how to do whatever we have to do in the US, including, health insurance. Like nobody knew
Yeah,
that you have your health insurance at work, or how it works. You also have, because, we use, I mean, I know that in France, I was used to have the health, dental, etc., everything
yeah,
together. You have your vital card and you just pay with it, and you never like spend the
yeah,
money. But here it's different. You have to find your dental plan, you have to find your vision plan, you have to find your etc. So I help those people to get where they have to be, and not, get in trouble when they have an emergency and they have to bank
exactly.
a huge amount of money. Um, and at the same time, I met with a rep at Leica,
Uh-huh.
at Bani, and we were talking. She was demonstrating her new microscope, the Mica, and we were talking, and I realized that we don't have any, industry intervention at the University of Michigan. And she was talking to me about like the job opportunity they have at Leica, and I was like, oh man, that looks so good. Like, I would like to know more about it. And I was like, maybe I can create a workshop. Uh,
Hmm.
to help this, facilitate industry coming to talk to University of Michigan and postdoctorals.
Yeah,
So what I did is I created, and this is important to be able to be in position of leadership. When you're in a position of leadership, you have much more power and also more credibility, where you can create stuff like I did. I created a workshop called "What's That Job?" It's a workshop study where we invite industry, and they just come and talk about the job opportunity they have in their company.
yeah,
There is sometimes university people that intervene, like, I don't know, the head of research at Omega that came to the University of Iowa and tell us about his job. But this is the kind of thing that is irrelevant to us, because this is attainable for us in 15 to 20 years. Like,
yeah,
it's not really representative of what you're gonna do in industry right now.
yeah. When you
So
enter, yeah,
Yeah, so I told them I created this workshop called What's That Job? And the two rules are the job has to be real and available right now, and it has to be available and for preferentially for postdoctoral fellows.
Okay.
And now so we invited multiple companies. We had Leica, we had uh, BioLegend, we had Vector Builder, and they came with job titles and they explained exactly what you're gonna do if you apply to the job tomorrow, basically.
Okay. And those just to clarify, those jobs are jobs that are open currently at that company. And then you
Yes.
come in and then okay, wow, this is really cool. How did the idea to about the name? What's
What's
that
that?
job? Yeah.
Uh, I don't know. So I was with it was over when my wife was there. We were talking about something, and she showed me this website to make ads, and I was like, "Oh, that's awesome!" And I was like wondering like how should I call the workshop? And
Yeah.
she asked me basically what you're gonna. I don't know. We just gonna present job, and she's like, "Well,"
Uh,
and it just came like that was that job basically.
I love it. I love it. And I think it's important because you don't, if you haven't been in industry, you don't know what to expect. You don't know what kinds of jobs you can go into in industry. What does that look like? And as you said, unfortunately, universities in general don't offer this opportunity to have that connection,
Yeah,
to be able
and
to learn about it.
they're not they try to cover themselves a lot. So we're work right now with this workshop. It's a lot of politics, which I like, so that's fine. But we're working a very fine line where it has to stay in the professional development aspect of the workshop. It cannot
Okay.
become a promotional event, and it cannot become a recruitment event, which is we're right in the middle of those two because
Wow.
the workshop has to present the product of the company, and people are gonna be like, oh, this is an ad for the company. It's like, no, you need to know what you're gonna go work for, and that's their
Yeah.
product, you know.
Yeah. Yeah.
So
No, absolutely.
they have,
Absolutely. No, I think it's really valuable. And on our end, Dr. GPCR, we do collect job ads, whether it's industry and or academia. We are in the process of revamping our jobs page so that when people come, it's easier for them to figure out, is it an industry job or a postdoc or any other academic positions. But we also have our own chief matchmaker, and his name is Mark Schmeisel. And Mark is actually a recruiter full time, but he also started out as a scientist, worked in science, but on the business side and marketing side for a long time, and has been a recruiter for quite some time. Um, and then in addition to that, we have not launched yet, but we're in the process of launching a new podcast and a new, or I want to say not new, but complementary ecosystem to the Dr. GPCR ecosystem, which is going to be open to all STEM PhDs. That's
mhm,
called Life Career Job. And the idea is, well, the first thing we're doing is recording podcast episodes. I'd like us to be able to release podcast episodes every 2 weeks, where we interview people who have transitioned from academia to industry. Some are consultants, some are working at companies, but really get an idea, a flavor of their track to where they are today, but also to get an idea as to what does their day look like, so
yeah,
that anyone listening can say, oh, I actually like to do this. I would be interested in doing this. Long term, the way I see the platform is, well, actually, it's the similar platform to the Dr. GPCR ecosystem. You register, we verify your identity, we let you in. There's an opportunity to talk into groups, but I would also like to have a life career job university similar to the Dr. GPCR University, where you can actually come and take a course about how to write emails, how to get ready for an interview, what to dress, how to dress up for an interview. Right now, a lot of interviews are on Zoom, but you can't show up in a t-shirt to a job interview, you know? You
Mm,
have to know, you have to get ready to answer questions like, tell me about a time where you had difficulty working with a coworker, but you had to finish the project on time. And then you need to know how to answer those questions. So the goal is really to bring in a bridge between academia and industry within the life career job. Because coming out of academia, if you decide that academia is not for you, you need a job. Ideally, you want to build a career in the context of your life, hence
mm.
the name. So very,
No,
very excited about that.
that's great. No, it is so important to have. We need information. We really need information
Yeah,
because it's so. We had this. We know the rumors, which are industry is evil. If you go in there, you're stuck. You cannot come back. Those are not true at all.
agreed.
Uh, we had BioLegend 2 weeks ago,
Hmm.
and the lady that came to do the presentation, Kim Cardenas, she explained to us that if you go to like biology and you can either do a job that I do where you can go on the road and help people troubleshoot their assays or and have a little sales part, or you can also be a scientist at the bench and you can publish.
Yeah,
That's something people don't realize. You can still publish when you're in the industry. It's
yeah.
different, but it's still publication with your name as first author. So
Absolutely, absolutely. It becomes less important to publish, but you can. And I think companies understand that. They like it when they get to publish scientific articles because I think it shows to a larger audience, to a larger scientific community that the science that they do is real science, just as you would do it in industry. So this person was an application scientist.
She was a field, no, she was a field scientist manager, so she was overseeing
Okay.
other people.
Okay. So yeah. But I think yeah, and you if you've never been in this position to talk to these types of people, you don't know what they do. What
You
does
know,
that look
and
like?
like through this workshop, I encourage people to volunteer for this workshop. What I tell them is, I'm gonna give you, you choose whatever industry you want to invite, and your role is to get in there, find someone that wants to come, and bring them here.
Yeah,
Through that way, you network like crazy. You meet everybody in the company, not everybody in the company, a lot of people in the
a little bit.
company
Yeah,
know your name. And if you are actually interested in applying for the job right after, I mean, they will know who you are. And that's,
exactly. That's I think that there's a lot of value in there, in doing that. And this way you're reaching out to people without asking them to give you a job. You just want information. And a lot of companies are very much interested in getting to know potential candidates, potential customers. You
yep,
never know.
that's, it's a
You
give
never
and
know.
take. We get information, they get exposure, basically.
Yeah.
And it works for everybody. And, and some people, you can do a lot in science. A lot is hidden sometimes, often, and you don't know it exists. And one of
Yeah.
them is art production.
Yes. So show us. We're
Yeah,
interested
I'm
in.
gonna show you. So I love to draw. I'd never have the opportunity to do it, but here is the. Oh, here we go.
Yeah.
So see, I designed those little guys. So our mascot is this little brain,
Mhm.
and basically I do a poster for every workshop we have, and
I love it.
it also those drawing also interested the people that intervene in our workshop. They're like, oh, who did that? I would love to like talk to them. And it's like, it's me.
No, I think this is wonderful. And then the other question I had is, how do you advertise these? Are these in person only?
Yes. So the thing is, that's where you have to be in a position of leadership. So find every university have some for postdocs. So find one and go for it. We have, as a chair of international affairs, I can justify it being professional development for postdocs and use postdoc mailing lists. and the university has to give us an updated postdoc mailing list at all time. So basically, I'm able to reach everybody. And then I also partner with different departments at university, so they do also internal ad distribution for me when I have a speaker. Let's say Pfizer is gonna come, I'm gonna ask we are partner with the Department of Pharmacology. They're going to provide the room for free for us to have the workshop in. The company is bringing refreshments. That's the only thing they're allowed to bring, food and drinks. They bring refreshments, and we bring the organization, and they do the advertisement in their department. So, it's really fun to build up a workshop from the ground up because you learn that you need to have pieces. So I need to have someone in the pharmacology department that I can relate to, the physiology department that I can
Yeah.
talk to, etc. It is very fulfilling. I really enjoy it.
You can tell. I can. It comes through the camera here. You can
Good.
absolutely tell. And when you think about it, the moment you decide what you want to do, these skills that you've acquired by creating these workshops, by networking, are absolutely transferable skills. You have to mention this. And then you could also think about how many people you invited, how many people you connected with, how many people came. And then very easily, that bullet point in your resume doesn't say one thing without numbers, but here you can say over a period of X number of years, you have from the ground up created this workshop where you had X number of attendees, X number of invited people, and it allowed you to develop, for example, relationships at the university and beyond across multiple companies. And then the multiple could be a specific number. And
Mm.
that, no matter what job you're going for, this already shows leadership skills. It shows that you're a self-starter. You're an artist as well, with a little brain with the hat, which I think is really cool. You could even think about whenever you do these workshops to get a t-shirt that has the just the logo.
I might do that. I have one printed for the workshop. I usually wear that one when I represent the university. So,
Yeah, yeah.
but I have one with the little brain.
Yeah. No, I think this is really cool. No,
No, for sure.
absolutely.
And yeah, and everybody tells me those are excellent skills. Like, don't forget to put them in your CV. It's gonna help you. Like, and
Yes.
this is the kind of thing that nobody's telling you to do, but you have, and
No.
you doing it proactively shows a lot of good competence that everybody is looking for, basically.
Yeah,
And
one other important thing is that yes, you're doing it proactively. Yes, nobody told you to do this, but you also have the support of your PI.
yes, that's because it takes a lot of time. I would say like my research decreased by an easy 35%. And he's totally okay with that. Every time, the thing is to be transparent, right? I'm not doing that behind his back. Every time
Yeah.
I do something, I'm like, do you think I should do that? I was on the search committee for the director of a new office at the university. Never done it, didn't know what to do. And I was invited to be on the search committee. So I asked my PI, should I do it? And he's like, I mean, yeah, you should do it. And it's good for you. Yeah, but I'm already like doing a lot of things, so is it going to be a lot of time? He's like, yes, it's going to take you a lot of time, but it's good for your professional development to do this kind of
Yeah.
things.
Yeah.
And but it's very nice to have a very supportive PI like that.
Yeah, I think it's very important as well to, as you mentioned, to be upfront and then to make sure that you can balance both things. Because at the end of the day, you still need to publish, you still need to get your papers out, your work moving forward. But then this is a great opportunity and a great time to develop your other skills.
And and also like it brings up my energy for doing research because research, after 8 years, is kind of like a job now. Even though I love
Yeah,
it, but it
yeah,
feels like more like a job now. And
yeah.
having
But
this side
yeah,
thing that I really enjoy makes me pump me up for research.
yeah, yeah. The nanobit assay is still a nanobit assay.
Yeah.
Up until the time you get to find the molecule or you're looking for that, answer, it's just quote unquote the nanobit assay.
The worst thing for me is transfection. I can't anymore. I every
It's
day.
yeah, I don't know. I haven't done that in a while, so I can't comment on that. It's been 3, 4 years now that I haven't worked in the lab.
Oh, this is the worst.
Yeah. Well, if you go to industry, depending on where you are, you can have a group that does that for you, you
Yeah,
know.
and I'm learning more and more about industry job, and honestly, I'm like 50-50 now. I don't want to be a PI. I'm a very poor grant writer, in my opinion. Uh, so I don't think I can be a PI. The idea of having to sustain a team with my writing capability
I
gives me
feel you.
anxiety.
I felt the same. I
Uh,
felt the same.
so
No,
probably not.
but it's a good thing as well that you know what you don't like or what you feel like you're not good at, because then it
Yes.
allows you to make the decisions according to your preferences and to your self-knowledge.
Otherwise, you fall into I don't know if you're aware of the Peter principle.
No?
You get promoted where until you get incompetent, basically. Your last
Oh,
promotion, and you are incompetent at your job.
wow. All right, let's wrap it up. Not that I don't enjoy chatting with you. I think we could be chatting for another couple of hours. Top three aha moments that you had as a scientist and that shaped your trajectory.
Three.
Three. It can doesn't have
Uh,
to be three, but anything that you feel like got you to where you are today. You know, the kink in the road.
hmm. One of them is definitely when we first used the nanobit assay, when we actually got it to work. Back in the day, it was a prototype that Julian is excellent with working with industry. So he got Promega. So Promega is the one that
Yeah,
commercialized nanobit. He got Promega to collaborate with us, and basically we got the prototype version of nanobit, and we were able to use it before everybody. I think there were like 2 or 3 other labs that was able to use it. But we made the beta arrestin recruitment assay through that collaboration and published with it. We were even, I think Promega allowed us to like determine what name we wanted to give the pieces and stuff like that. It was very, very fun. Uh,
nice.
don't quote me on that. Well, no, it's fun.
Maybe it's
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 fun,
it's
but
it's how you remember it. Who knows?
yeah, exactly. I remember that we had a lot of freedom, and it was a great experience. And when we got it to work and we could see the actual dynamic recruitment to the GPCR, it was a great moment. My second one is definitely the gate model. Uh, when and especially that I got a little bit of pushback on it because
Mhm.
I understand I don't have any proof, but it's still a hypothesis,
Yeah,
and I think I had enough data to state it. But I got a little bit of pushback on it, and when and Julian was with me when we saw the even though so when we saw the I still don't remember his name. When we saw the talk of the guy at the Mayo Clinic that did the same discovery as me, uh,
gonna look it up while you talk.
yeah, oh yeah, I look at Julian and I'm like, so apparently it's not nonsense, is it? And we hadn't published the paper yet. The guy at the Mayo Clinic published it, and but I published the thesis with the language in my thesis.
Okay.
I don't think Julian saw it, so it went through smoothly.
All right. So this was the 20 Niagara on the Lake, right? In
Yes.
2022. I just logged
I,
in. Oh,
yep.
Scott Blanchard, of course. Yeah, yeah, yeah. Oh, actually, it's so interesting
Oh,
because I
St. Jude Children's Hospital. I'm sorry, it's not Mayo Clinic at all. I'm so
yeah, yeah,
sorry.
yeah. St. Jude. Yeah, yeah. Scott, I think yeah, I think his talk was really phenomenal. And him and I were actually at the end of in the back of the room right before his talk. And we were chatting. He's a wonderful guy. Really, really awesome.
Yeah, I mean, you see, like once again, I didn't even talk to him when we were at the conference because I was still in my introvert phase. I only grew out of it after that, but I emailed him.
He's wonderful. He's wonderful. And it was interesting. I do remember his talk because One, it was a great talk, super nice guy, but also in his talk, he had mentioned that his lab develops tools and he's not a GPCR guy.
Hmm.
And he blew I mean, the talk was phenomenal.
It was very good. I
Oh,
agree.
that's awesome. All right, that was my last question. Alix, thank you so much for being here. Don't go anywhere. We're gonna let people wonder what we talk about afterwards. Just kind of a debrief. But thank you so much for your time. It was really cool talking to you.
And thank you so much for having me. It was fun.
Thanks. Can I ask you a favor? Please subscribe to our YouTube channel. Many of you come back and watch our videos but aren't subscribed. Having more subscribers will help us get you more content. Also, mark your calendars and join us for the inaugural Endocrine Metabolic GPCR's meeting taking place April 22nd to 23rd this year in Birmingham in the UK. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guests, our Dr. GPCR team members, Attila, Ines, Montserrat, Ivana, Andreina, and Balint. If you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials and subscribe to our monthly newsletter directly in the ecosystem. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Don't forget, we're also starting up the Dr. GPCR University. So if you'd like to join us for Dr. Karen Atkins class, or you'd like to teach a class, please don't forget to reach out to us at hello@drgpcr.com. Until next time, stay safe.